All right, let's get him back and try to start. And so we will have a course review now. And again, the final exam is December 2nd. I haven't made any exam question yet, but here is the knowledge point I want you to understand. And during this review, if you have any question, including is this going to be in the test? And if I cannot tell you, I'm going to tell you I cannot tell you. But you may get lucky that I say this won't be in the test for some of the things. So just feel free to stop me. And this is actually, if you print out the things, it's very light. It's not heavy at all. The first the concept that we had is, of course, what is next generation sequencing, right? It's sequencing and it's short arrays. I think at this point, I hope you all appreciate that. And it's ultra high throughput. The data amount is huge. And as you can see, I only sequence the beginning part of the sequencing or both two ends of the sequencing. That's called the pair end or mid pairs. Okay? So basic concept of next generation sequencing. And we talk about many different type of platforms. And this is something that I expect you to understand too. And the leading platform is the Illumina and Solid platform. For Illumina platform, and the amplification is the bridge amplification, if you still remember those things. And the sequencing part is the sequencing by synthesis for the Illumina platform. For the Solid platform, and the amplification part is called the emotion PCR. And the sequencing part is the sequencing by ligation. If you still remember how they designed those probes and then eventually add up another couple of add up another couple of things. Another interesting feature, most important feature of Solid is the data doesn't come straightforward as ACGT, rather it's a color space. And every two dinucleotides, they will have a color. And so AACC, GGTT, they are all the same color. So make things a lot more complicated. But the added benefit for that is it's more accurate because every single sequence reads, you actually sequence it twice. You read it twice. So if the sequencing error is 1%, now by Solid, in theory, you got 1% of 1%. So it's much more accurate. And another platform, I think is going away. That is actually the first platform that was commercialized, which is the 454, and based on the pair sequencing protocol. But the most important feature is it's long arrays. It has about 400 base pair arrays, and can do a lot of things that are interesting. But some people that you were in the Aaron Torrent seminar, that you can see that Aaron Torrent is pushing towards the same direction and in about the same scale. Right now, they are at the stage of 200 base pair arrays, and they are going to have 400 base pair arrays very soon. And the cost of that is probably 1/10 or less than 1/10 of a 454 technology. So I'm thinking that 454 may not be lasting too long. And another platform is the Pacific Bioscience, and the major importance of that is the single molecule sequencing, which means that you can skip that stage of either bridge amplification or emotion PCR, which both of them will potentially generate a lot of errors. And then another very important fact is it gives you very long arrays. It's about several kilobase arrays, and that is good for a lot of other applications. Okay, and Aaron Torrent is a personal genome analyzer. That's how they call it. Apparently, I don't feel very appropriate if they call it this way at this stage, but and that is the first sequencer that doesn't have that kind of expensive camera to take the pictures. Rather, it's using this ion-sensitive semiconductor technology, which is very, very smart. Cheap equipment, lower throughput, and higher error rate at the moment. And so far, I think the error rate is about less than 1%. Their technical report is 0.1%, but I think it's over estimate. And different applications with the technology, and this is actually the backbone of the entire semester, right? So we talk about for the sequencing, for DNA sequencing part, and the basic importance part is the reference-based sequencing to identify genetic variants. And for the RNA sequencing part, for the ChIP sequencing part, and for the epigenetics. So this is one slide summarizes what this technology is capable of. Still no questions so far, right? Perfect. And a very important concept, and I think I need to have a question on this, which is the pair ends and the mate pairs. And I know this is something that is just the information that, but once you read the papers, once you see the pair ends or mate pairs, if after you learn the next generation sequencing, you still cannot distinguish the difference between the two, that's something not very good. So I have to force you. I will have one question on this, okay? And so for both of these two technologies, and for the Illumina and Solid, and there's a single end, so most standard operation, and the Illumina can get to 100 base and Solid 75 base pair, and this is a fragment length, so you can see the subtle differences. But for the pair end, and we know that the fragment length is pretty short, it's about 200 to 500 base pair, but you still sequence both two ends in this direction, and this is for the pair end for the Solid protocol. But for the mate pairs, the major differences is the length between these, the DNA fragment. This is a couple of 100 base pair, but this is 2 to 5 kb. And this is in the range of kb as well, so this is pretty long. And in terms of the sequencing directions, they are a little bit different. So, and you can go back to have some study. You don't have to remember anything. I just expect you to understand it, right? Because this will be in the slides and you'll be able to see it. No questions about the first lecture? We're all good? Yeah. Why pair end sequencing is good for RNA-seq? So the question is why pair end sequencing is good for RNA-seq. So it depends on what's the biological question for the RNA sequencing. If you only are interested in the gene expression levels, probably single end is as good, right? But if your goal is to identify the alternative splicing or gene fusion, and if you sequence both two ends, that gives you a lot more information that you can work with. Okay? Some of the DNA fragments, they are not that long. If some of the transcripts, they are less than 2 kb. So you can see this fragment length here is it's about 2 kb long, 2 to 5 kb. You might miss a lot of things if you use mate pair. Yeah, I wouldn't say you cannot, you still can, but probably you're missing something. All right? Are we still perfect? All right, we're still perfect. And another thing is when you work with RNA, there's a lot of degradation happens. And you're assuming that the RNA molecule is perfect, but sometimes it's not. So even though the initial transcripts are pretty long, you probably still only work on very small ones. And so I probably mate pair won't work for the RNA sequencing. Yeah.
You're probably not that far apart either. I mean, really that useful.
Right, but you're trying to sequence the transcript, so there's a combination of multiple exons. But anyway, mate pair won't work for RNA-seq. If you have a question on that, and we can plot something after the class. And any other questions here? No? Perfect. The second lecture was really the basics for data processing. And this is most on the secondary analysis part. If you still remember what that is, it's not specifically to any biological applications. Rather, it's as long as this is sequencing, and this is something you have to do, right? And the first concept that we gave is the 3 qualities, right? So when we talk about, it can be really, really confusing when you talk about the quality of the data. And if people ask me, what's the quality of this sequencing data? I really don't know how to answer that question because there's so many different definitions on the quality. And there's 3 levels of quality that we talk about that we can have very solid evaluation on that. The first one is the so-called base quality. That means it's for each individual single read. You've got A, C, G, T, and things. Let's say the first one is A. What is the quality of that? How confident I am that is A, not a C? Okay? So for each nucleotide you actually sequenced, there will be a quality associated to that. That is called the base quality. Are we okay? And the second quality is called a mapping quality, which means that for each individual sequencing reads, when I map it back to the reference genome, okay, it can always be mapped somehow, and it just depends on how many variations in that. And what is my mapping quality? How confident I am this is the right place to be. Okay? And so for each individual sequencing read, I'm going to have one mapping quality. The third level of quality is the consensus. Some people call this variant quality. So consensus quality. So for each individual genomic loci, if I say this is the same as reference genome or not, if I say this is a homozygous, heterozygous, or whatever that is, how confident I am for that. So this is not really associated with independent read. Rather, it's for each genomic location, I'm going to have a consensus quality. Any questions? They're still perfect? They're still perfect. Good. So, and another very important concept is when you evaluate the qualities, and people don't really give you the p-value, because the p-value is not a really nice number to work with. You say my p-value is 0.000001, or you just tell them I'm really confident, and that will be fine, right? And you need a lot of computer resource to remember that. So what people use is called Phred scores. So basically, they will convert the p-value into this log base 10 and time 10. So if it's 0.01, which many biologists use a standard, and your Q is actually 20. If it's 0.001, your Q will be 30. So this Phred score is something that I may give you a test on. And so once I say I'm going to give you a test on, and I see somebody start to make notes. And if I don't say that, and people just listen. And people start to ask questions. Yeah, no, actually, these scores are associated to all different 3 types of qualities, and people are used that. And it's just if it's base quality, and that will be reported by the sequencer, and if it's mapping quality, it will be reported by the alignment algorithm, and this can be reported by the variant colors. You good? And we spend quite a few time to talk about alignment algorithm, and start from Smith-Wortman, and which I know I didn't do a good job, but there's no way I can do a good job in about 10 minutes to talk about Smith-Wortman. But just to give you a flavor of how dynamic programming really works, but point is it's not going to work for the next generation sequencing data. There's one more concept I also want to mention is when we do the alignment, when we talk about alignment, there's 2 concepts. One is a global alignment, the other one is a local alignment, right? So you don't have to think hard to make a conclusion that for the short read sequencer, the sequencing alignment is always a local alignment. It's always local alignment. And the major challenge of the next generation sequencing data and alignment is going through 1 trillion times of dynamic programming is practically not possible. And the features of data, they are short and they are massive amounts. Okay? And this is a summary slide to tell you what's the considerations when you talk about when you select different alignment algorithms. And the first, of course, is the cost, right? It's the cost meaning the speed and the computational resource that is required. And we know probably in terms of cost, the bow tie is the best one, which is listed here, right? It's very fast. It only uses, I think, 3 gig of memory to align for the human genome. It's something like that. But anything that comes with a chip that has to have some other part that is expensive. So you have to sacrifice something. So there's more information you really need to consider, which is including the information being considered, meaning that when you do the alignment, whether the base quality has been included or not. Some aligners did, others don't. And alignment quality, this is the most important thing that I want to stress on, which is some of the alignment algorithms, like a bow tie, doesn't really allow you to do align gaps. So we know that once we kind of sequence 50 base pair arrays, we map back to the genome. And if there's 1 or 2 base pair gaps in the middle, that is small insertion deletions, it won't be aligned in the bow tie. And if it's a mismatch, that is OK. If it's the insertion deletions, it won't be mapped. And that is how the index and how the algorithm is designed. So this is a big problem for bow tie and for many biological applications. Another thing is you want to consider is the accuracy, which is the sensitivity, specificity evaluation. And you are probably out of luck on this, and there's it's something hard for yourself to do the evaluation. But there's some people do some review papers, and they compare different approaches, and actually they use different, they design their own benchmark data and to test the different algorithms. So those are the things that you need to pay more attention on. And another thing I forgot to put here is in terms of consideration is some aligners can be worked on the color space data, and others cannot be. Okay? And if you're working on Illumina data, that is okay. It doesn't matter. But if you work on solid data, and the color space consideration is one of the major things that you have to think of. And some of the algorithms, actually, you can do color space, but for color space and base space, their performance are different. So we have one of the paper talk about, I think, comparing BFAST, BW, and bow tie. And we know that, yes, BWA allow gaps, and it can be used to work on the color space, but the performance is way worse compared to the BFAST. Of course, BFAST is much slower. But those are the things that you need to consider when you pick the different alignment algorithms, right? And in terms of alignment algorithm, that I'm not going to, this is something I want to make sure. I'm not going to let you to do the Bolling-Wheeler's transformation in the class, in the exam, okay? I know that probably will kill you, and it will kill me as well when I grade it, so I'm not going to do that. But these are some of the information that I want to deliver is for the alignment purposes, we started from Smith-Wortman, that is the dynamic programming for the local alignment. And for the fast alignment algorithm, there's a hash table based and surface tree and previous tree based. And here, these are the things, if you go back to your lecture notes, and these are the computational resources, at least in terms of the memory usage for different type of strategies. For this is the BWA based, and you can see this is bow tie. It's only used 1.65 times the genome size. The genome size is 3 billion letters and 3 gig. So this is very small, and most of the current laptop computers can really do this. And for this type of thing, it requires 45 times, and that is really big. And what we are using most is somewhere in the middle, and not as small as this one, but and someone that is used more. What my group is using is the BFAST, and it's pretty. It's okay in terms of speed. It's pretty slow, but we can wait. And the memory usage, it takes about 32 gigabits of memory, and for us to run. But if you use a smaller size of the memory, you can do some other tricks, like breaking the genomes into smaller pieces, and each piece is kind of small, and then eventually you combine the data together. And that is something that we are using right now. And after the alignment algorithm that we were talking about, the file format, and including the initial raw data is the FASTQ files, okay? Try to go back to see what that looks like. And for the SAM and BAM files, all right? So we have, and for the SAM and BAM files, there's many different fields in there, okay? I'm telling you, I hope you understand those file formats as much as possible, but I want you to understand at least how to interpret cigar strain, okay? I know probably 70% of you don't know what that is, and at this stage, you probably never heard about it. Actually, you did, but you just don't remember it. And I want you to know about that, and I will guarantee I will have a question on that, okay? And the next format is the VCF files. This is the documentation for the variants, and I expect you to understand most of the fields in there as well. Okay. How are we doing on the DNA sequence for the lecture 2? The basics, the alignment, and the file formats, nothing else. Are we still okay? All right. So let's jump to the next topic, which is the DNA sequencing part. We had 3 lectures on that, and the lecture 4 was the lab tour and the biochemistry part for the library preparation. We'll have 20 points question on that. What are you laughing at? No, we are not going to test on that. I know you don't have slides on that. And I wasn't in the class even, so I don't know what was talked about. But lecture 3, 4, 5, and 6, they were all on the DNA sequencing part. And the first knowledge point is the types of genetic variants. So this is not really related to the sequencing, but this is some basic biological concept I want you to understand. SNP. What is SNP? Single nucleotide variants, right? Polymorphism. And I want you to just keep in mind when you read that 1% of the population needs to have it to qualify for SNP. Otherwise, it's called a single nucleotide variation. Not terribly important, but you have to see this is SNP is a population-based concept. And I want you to understand transition and transversion, okay, what that really is. And there's other type of genetic variants. It's short insertion deletions, repetitive elements, and large structure variants. And those are different types. And we talk about the GWAS. And so I'm not going to go very detailed on that. And so basically, GWAS, genome-wide association studies, is the common disease, common variants, and assumption. And so you can see this X here is allele frequency. And towards the right side of this, that is more and more on the common variants. As you can see, this is 5% of population have it. This is 0.5%, and 0.1% have it. So towards the right side, this is common variants. But this is Y here is the effect size. So meaning that some variants may be very rare, but once you have it, you're dying. That is a pretty large effect size. And so for the GWAS, so basically people focus on this part and the common variants and the common disease. And rare variants, basically people use a lot of family-based studies into trying to figure out that part. And the next generation sequencing part, especially the exon sequencing, and help us to go through this part and somewhere in the middle. I want to make sure that we will have questions from Tatiana. Okay, I'm going to ask questions from him. Haven't have that yet, but I was in the lecture, so I know what she talked about, and we do have the slides, and so make sure that you review her part. And so there's the limitations of GWAS and all those things that you can go back and to see, and why the next generation sequencing can help with that. We talk about a couple of examples, and the first one was the whole genome sequencing example, and the exon sequencing and the pooled sequencing. So all those things, just basic concept-wise, and I hope you can understand those. And I think this is somehow related to Tatiana's part, and when she talked about the family-based structures and exon sequencing, and hopefully that you can answer that question pretty well. And we also talk about the personal genome sequencing part, and this is not really on the research part, which is identify the genes that respond to certain disease. This is for the application. So the first one we talk about is a 40-year-old man that with a family history of heart disease, but this is a general healthy person. And then this goes to the clinic and have a lot of diagnosis, and everything looks normal, but some of the factors indicating potential disease risk. And then after having the sequencing information kick in, that can help us to improve the clinical diagnosis. So that's the point of that. And we also had a story of this kid that had a disease that nobody knows what's going on. And then they just did a very simple exon sequencing on this kid, and they find out which gene caused the problem, and they fix it using cord blood transplantation. Okay? And this is another concept that I expect to understand, the scope of their DNA sequencing. And we talk about the whole genome sequencing. There's a couple of examples: the Watson Genome, Southern Genome Project, Cancer Genome Project, and different strains of animals. Okay? And at a time that we are not that rich, and people do capture-based resequencing, and including the exon sequencing, right? When Tatiana's question comes in, whole exon sequencing project. And this capture technology, we can use PCR-based assay, capture array, or capture in solution. And the region selected can be only focused on exon region, or can focus on certain regions that you already have previous biological knowledge. And if we are even poor, we cannot even afford for the capture-based sequencing, and some people do the pooled sequencing, that the goal is to identify genetic variants. So some of the concept here, I hope you can review that when you're studying this. And for example, what is the goal of the pooled sequencing, right? What's the difference between that with the exon sequencing? So some of those, I want you to understand the concept and go back to figure out. Once we talk about the analysis part of it, and the first concept in terms of informatics is a mappability issue. And we know that human genome are repetitive, and so non-unique mapping can really cause a lot of analysis errors. And one question that we had is what the read length is, and then how that corresponding to the mappability. So we know that, remember we had one calculation, we calculate that if in principle, every 16 base pair, you should be able to find a unique genomic location if everything is random. But now human genome are many regions, actually 52% of human genome are repetitive regions. Only 48% are non-repetitive regions. And so for one of so if you use the shorter arrays, it really doesn't give you a great answer. But once you increase the length of the read, and you increase the mappability. So you can see here is, this is for different species, and human is the blue part, which is somewhere here. This line is the human. And that tells you that around the 50 base pair, your mappability is about 80%. 80% of the genome are mappable. Further improve that read length probably won't help as much. It will still increase a little bit mappability, but not as much. So here comes the question. Why that I'm talking about human genome, 52% of human genome are repetitive regions. But why this short arrays can really give you 80% of mappability? Okay, and I'll give you the answer for that. Is if this is one region that is repetitive, and this is the sequencing rates, if it completely locates in the repetitive region, you are not able to identify where they are because there's so many different regions that have the same one. But it happens to be on the border part of it, and this part is unique. And then that can help you to get back a lot of regions that you otherwise cannot find out. And this is one of the things that when we talk about the tiling array versus sequencing. Tiling array, 52% won't be on the array at all. But for the sequencing, you can really having a much broader range. You can recover over 80% of the genome. Any questions so far? There might be a question on this. I don't know yet. Mappability is region-specific and alignment algorithm-specific. If you don't know what that is, go back to your lecture notes. The second topic when we talk about DNA sequencing in terms of informatics is realignment, refined alignment. So this is a little small here, but you can find out in your lecture notes. And some people find out that if for some region that there is an insertion deletion occurred there, that will increase your error in terms of mapping dramatically. And those really, sometimes it gives you the artifact that that is a variant. Actually, it's not. So this is one example that before you do refined alignment, it looks like this. You are looking at here is two sequencing variants that are very close to each other. However, after you do the refined alignment, you don't see anything here. It's just a couple of insertion deletions that cause those sequencing error. And sometimes that is really misleading. And comparing to initial alignment, the refined alignment actually minimizing mismatches for multiple rates versus one rate. The initial alignment find the best mapping for that one rate. For the refined alignment, it's for multiple rates. That's the only differences. And it's a two-step process. And the first one is to identify suspicious regions. There's a couple of strategies for that. I know you don't remember that, but go back to the study. And the second is actually conduct this realignment algorithms. And this include identify the haplotypes and then conduct gapless alignment on all haplotypes and calculate likelihood. So I know this won't make any sense if you don't study the lecture notes. And the third topic we talk about, the computational part, is the base quality recalibration. So the basic point here is all the algorithms, all the different platforms, they lied, right? So this is especially for Illumina. So you can see that X here is their real quality. No, this is reported quality. And Y here is the real quality. So you can see once you go to the downwards of this 45-degree line, that means the reported quality is not as good as it should. The reported quality, okay, the real quality is not as good as the reported as. So you can see that they lied. Anyway, so those higher than the real quality scores provide false confidence in the base cost, and sometimes it can be 10 times, and it's very dramatic. And the recalibration part of it using multiple features, including the base quality and the alignment things and the sequencing context, and so you can go back to take a look at what's the information people use to really make these corrections. And the fourth topic is the variant identification. So if you identify one, this is a reference, and there's so many sequencing rates along this region, and whether this is a variant or not, whether this is heterozygous, homozygous, or it's not a variant at all. So, and it depends on the experimental design. And for the deployed genome balanced design, there's one approach. But for the unbalanced design, there's a different type of interpretation, right? If this is for the balanced design, deployed genome balanced design, this is going to be a G, homozygous. And the A is probably an artifact here, although the reference genome is A. And the only reason for that is we are expecting half from allele, the first allele, half from the second allele. they cannot be that imbalanced. And however, if for cancer genome, if we see this, we probably will have a different conclusion. The major reason for that is for the cancer genome, sometimes it has mutation. A small percentage of cells have that, and others don't. So you can see this, and it still makes sense. For the pool sequencing, if only a small percentage of people in the pool have it, and you still can see this. So A is still real. And for the RNA sequencing, it can be caused by the allele imbalance between two alleles expressions. And variant identification based on sequencing from multiple individuals, there's another type of, but you don't have to, if you're interested, you can look at it. I'm not going to test on that. Any questions so far? So this is a variant identification. And variant quality recalibration, this is another thing that it can be very interesting as well. Because once we got variant quality, when we say this is a variant here, okay? This is a homozygous. And there's many different measurements on that as well, and including the strand bias, including a couple of other things. But is that real or not? And what people using here is they will use a machine learning approach. So you got all this measurement for one variant, and then you use gold standard and trying to train your machine. So once new data comes in, and then you just recalibrate the probability of whether that variant is real or not. The GATK package can help us to do all these things, but you have to understand what's the basics of that. SNP quality evaluation. So the point I want to understand here is for different type of study designs, and there is actually kind of, you can expect the number of variants you identified. If you do data exon sequencing and you identified 1 million variants, that's not going to be right. And so these are the things that you can really see the expectation how many variants are there, and this is after you identify those variants, this is the first thing that goes through your mind. Is that number looks like right? And here are the numbers that you should be considering. And SNP quality evaluation, all right? So there's a couple of things. So once you see there's a SNP, or there's a bunch of SNPs identified, is that real or not? So what the first evaluation is that people consider as concordance with genotype ChIP+, because for a lot of samples, we already have the SNP arrays, okay? And if for those samples, you can really see whether that is agree with what the array type of technology called for. And but more realistically, most of the studies, so if we don't have the ChIP count, the first one cannot be used. But the second is important. Is what's the fraction of SNPs are already known? Okay, so if you identify 30,000 variants in the exon sequencing, and you want to see 90% or 95% of it has been identified in dbSNP or in Southern Genome Project, you are probably around the ballpark part. It's probably right. If you see only 2% are documented in human genome, you want to make sure you're still sequencing human. And at least the very least use the same genome build. And there's something wrong with it. So look for the fraction of SNPs that are already known. That gives you a good indication whether the variation identified is correct. And another very important part is that's why that in the initial slides I asked you to understand what is transition transversion. And transition transversion ratio is a good evaluation. I'm not going to go detail, but that's something you want to really study. I guarantee there will be questions on this. Okay? People start to take notes, I know. And the structure variations, again, there's the different structures, large structure variations. And we actually talk about a couple of methods that identify structure variations. The first one is methods based on tag density, and the second one is a method based on pair end mapping. And these are something that we don't do it every day, but I feel like we have to go back to do it because there's a lot of exon sequencing data now. We do have the pair ends, And we are not really using those information if we don't do this type of structural variation identification. Those are something that I expect you to understand as well. And the bioinformatics approach that related to the DNA sequencing, so these are the general major components in the analysis: raw data, base quality recalibration, alignment, refined alignment, variant quality, variant recalibration, and then the next part will be the functional prediction and the rare allele association. And these are further on I think it was on the third lecture of DNA sequencing part. And of course, after that, we'll need to have further biological validation on that. So there's one slide that I'm going to talk about this part, which is the function prediction. Okay, and there are different strategies on that. And after you do the alignment, refine the alignment base calling. And the first thing you want to do is the annotation, whether which variant is in the intron region, exon region, or whatever. Whether it has been documented in dbSNP 1000 genome. Okay, if it is, it's less likely to be important in the disease because the 1000 genome samples, they are generally healthy, and it's unlikely to be in the disease cohort you're studying. And we talk about molecular functions, which are one of them are changing protein structures or affecting splicing and gene regulation. So we spend quite a few time on that and take a good look at it. And for the statistical analysis part, and that will be the disease association part. And for the individual variants and based on disease model, so we talk about the dominant model, recessive model, and this, I'm hoping that Tatiana will put this information into her question. Family structure, whether that can help and control and the case differences. And next one is the collapse of multiple variants in one gene. And these are something that so far is still under fast development. So I'm not going to have any question in here regarding the final exam. And for the variance prediction function based on the molecular functions, which is this part, the molecular function, the annotation of molecular function. And we spend some time talking about the non-synonymous variants. Those are most people are really focusing on, and they use basically the evolution conservation, right? Comparing different species and how conserved regions are. More conserved, more likely it's biologically important. So people do the structure-based or sequencing-based prediction, but all based on the conservation. And we spend quite a few time I was trying to justify why synonymous variations are also important. And these are different things that I'm not going to reiterate, but it has something related to post-transcriptional regulation and translational regulation and protein stability issues. So there's a variance especially in the exon sequencing in the exon regions can be important in the regulation part, not only changing the amino acid sequences. Are we still cool, or everybody just got so confused? If you are still confused, go back to static lecture notes, okay? So that's the DNA sequencing part. And now let's come to the RNA sequencing part, the knowledge points I want to have here, okay? And the first one is RNA sequencing, transcriptome sequencing. It's more than gene expression. So in the microarray world, we all only study gene expression, but that's only one-dimensional thing. And actually, for the transcriptome variation, it can be splicing differences or can be transcript variants and variations on the transcripts. And RNA sequencing can help us to addressing all these issues. Well, microarray can only measure the gene expression levels. And we talk about sample preparation part. And so these are considerations, that the amount of things required. The ribosome removal, remember, that is a very fun topic. And we have to do it very carefully. Otherwise, you're only sequencing the ribosome RNAs, which you are not interested in. What is the reverse transcription method you are using? You say oligo-DT or random primer, because they are different. They capture different things. They have advantages and disadvantages. And whether your assay maintains the strand of the sequencing of the transcripts or not, they are different. and whether you want to do the pair ends or not. Okay, and Milan Radovich gave a lecture on this, and I will see whether he will provide one question on that. But certainly, I want you to review his slides as well. For published studies, and we go through those, and so basically the first RNA sequencing experiment and alternative splicing, gene fusion, single cell transcriptome analysis, genome annotation for non-model organisms, and various in the complex diseases and small RNA profiling. So those are the examples that is only enabled by the RNA sequencing technology. So this is what we can get out of RNA sequencing. So it's got RNA sequencing. You can study gene expression, alternative splicing, and the variants, how to connect those variants, predict the biological functions, and then make the connection back. And you can also study allele-specific expression and non-coding RNAs. Okay? And the first informatics topic we talked about in RNA sequencing was also alignment, because the alignment of RNA sequencing data is not that straightforward. And the major reason is for the sequencing reads that cross multiple junctions, most of the short reads aligners won't tolerate that. So those reads won't be really aligned. And for this type of algorithm, so there's two different types. One is the exon first method, and the other one is seed extension method. So I'm not going to go back to the details. So you can go back to study a little bit. So the exon first is you do the alignment first and focusing on only the junction, the reads on the exons, and then further look for the junction part. While for this one, for each individual reads, you're trying to work through and to identify the junction reads. Okay, not going to detail of this, and please review this part. For the gene expression part, and this is something called RPKM measurements. So the point here is how do we measure a gene expression based on the sequencing information? And you got one gene, and there's some of them express more, others not as much, but this is longer than this one. So the total number of reads is determined, should be normalized by the total number of reads and also the length of the transcript. And this is something called RPKM or FPKM if it is a pair-end sequencing. I'm not going to go through this one, and you can feel free to go back to this as example. But I will tell you, RPKM calculation is one of the knowledge points that will be appear in the exam. This is the exon union method and the exon intersection method. This is still looking for the gene expression level analysis. So if a gene has multiple isoforms, you can use two different strategies, and both strategies have their advantage and disadvantages. Please go back. This is one of the things that will also appear in the exam. Alternative splicing. This slide just tell you what is alternative splicing. I don't want to talk about that again. And the point here is for the computational strategy, there's an isoform-based approach and the alternative splicing event-based approach. For the isoform-based approach, you look for different isoforms. For the alternative splicing event-based, you only look for this particular event, which you don't care which isoform it locates in. And for the isoform-based event, you can do genome-guided reconstruction, and which is Cufflinks or scripture. Remember, we spent pretty much a lot of time on that. And there's a genome-independent reconstruction, which means you have to do the assembly first, and then to figure out your transcript variants. And this is for the Cufflinks and scripture, and the commonality of those are both needs to have the splice alignments. But the difference is I want you to appreciate that. I'm not sure this will be in the exam. I'm not saying it won't be, but I'm not sure how do I have questions on that. For the Cufflinks, here is you are maximizing the specificity, meaning that if I say there is something, I want it to be right. I may miss a lot of things. But for the scripture, it's maximizing sensitivity, meaning that if there is something, I'm going to report it. I'm not going to miss it too much. But For the ones that I reported, you may find more false positives on that. But this is a major difference between two approaches. And the MESO is approach that is exon alternative splicing event-based approach. Okay, event-based approach. So it only focuses on one single event, and it tells you the percentage of inclusion that in the final transcripts. It doesn't matter which isoform it locates in. It doesn't matter whether it's there or not. So just go back to study that a little bit more. Gene fusion. So what is gene fusion? These two genes doesn't supposed to be together, but somehow they end up with on the same transcript. And there's some approach that using pair-end arrays, and there's some approach using single-end arrays. And yeah, and you can probably go back to the different methodologies. And here are the methodologies. The red ones are the ones that we really talk about. And these are not really complicated, and actually there's only 1 or 2 slides for one of the algorithms. So feel free to take a look at that. Allele-specific expression, and so the first point is, what is that, right? So when we talk about the 2 alleles, their expression levels, when you study cis-acting regulation, they serve as a control for each other, okay? And because 2 alleles, they live in the same cell. And all the conditions are the same, except for the sequence variants appeared on those 2 alleles. And there's a technology study that we talk about the padlock-seq assay. Still read it, but I'm not going to test it on that. And for the RNA sequencing assay, and when I say I'm not going to test about that, some people get a pen, cross that off. So, but that is some cool assay and to understand. So feel free to see that. For the RNA sequencing, and there's some major difficulties, and all the alignment algorithms are based towards the reference allele, and it requires both DNA and RNA sequencing, and hundreds of reads per base pair may be required for subtle variants. By saying subtle variants is less than 1.5-fold differences. It's sometimes in many situations it's very, very difficult to get through that. And we also talk about the RNA editing, and it does exist, and it's controlled, and it's not random error. And RNA sequencing, if you do the experiment right, you can identify those. Okay? And I'm not going to go details on that. And the next topic will be the microRNA sequencing, right? So here is the sequencing itself. You've got a 22 base pair, and you put the linkers on both 2 ends, and then you sequence it. And the trick here is it's only require about 35 base pair arrays, and have a longer arrays won't really help, because for those microRNAs, they are only 22 base pair by themselves. So your sequencing, because of this, is too short, and the sequencing will run into the 3' adapters. And you have to have some additional algorithm to remove the 3' adapters. And we talk about 2 different approaches to deal with the microRNA sequencing data. The first one is more on the known microRNAs. This is the DSAP. I think this is from Tsinghua University in Taiwan. And the step 1 is to remove the adapters. The step 2 is cluster reads into unique sequencing tags. And the step 3 and 4, they were actually mapping those against the existing known database on the non-coding RNAs. But the most important part of this is for the de novo finding, de novo microRNA detection, because we don't really know too much about microRNAs. And this is the microRNA deep approach published in Nature Biotechnology. And they are taking the advantage of this microRNA biogenesis process. So you can see that we know this is a microRNA precursor and form this hairpin loop. And dicer really come here and to cut here, okay? Because of this, the pattern that we are looking forward is looks like this. So here this part is the microRNA part, and we'll see some signals about surrounding those areas. And if this is not a dicer-specific cleavage, which is unlikely to be the microRNA precursor, and then your cutting will become random. And then your sequencing, once you map reads back to this region, it will be somehow different patterns. So we are more looking forward to this type of patterns will give us more confidence that it's a dicer-specific processing regions and give you more confident that this is a microRNA, a novel microRNA binding, novel microRNA that otherwise you will miss. Are we good on RNA-seq? You still remember I talk about, can you believe it? We actually talk about all these things. And I don't even believe I still understand most of this, which is nice. And the last part that we went through was the ChIP+Seq, right? This is ChIP+Seq. I'm not going to go through that again. And we talk about a couple of studies on this, and the transcription factor mapping, that's very important, and the histone mapping, and this is also a milestone paper that in the ChIP+Seq, but this is on the chromatin structures. And for the enhancer identification, these are the two things that use the P300 as a marker. And study DNA methylation, and there's a very interesting review paper that probably you want to go into that if your research interest is this. Okay, I'm not going to tell you anything in this paper. So most likely you won't study it, I know it. And, but you can also study protein RNA interaction and study microRNA targets. Remember, if we do the ChIP+Seq, and then we target on the Argo protein, which is the one part of the risk protein complex, and people use that to really study the microRNA targets. That is really interesting application. All right, and the remaining things I think I already gave a brief overview today and in the beginning, so I'm not going to reiterate that. So this is the experimental considerations, and here is the class of signals in the ChIP+Seq experiment: transcription factors, RNA polymerase, and histone markers. And the tag distribution should look like this, and there's a positive and negative strands, and tag shifting approach, and tag extension approach. So these are the things you can go back. And sliding window strategy that can denoise the signals. And this is the multiple binding sites, I think. And for the last one was the fragment length evaluation based on the computational approaches. Motif finding, and these are the things that I hope you will appreciate and which are important. And some of them we will have them in the exam. All right. Questions? Still don't have questions? Okay. The exam will be on December 2nd, and I will try to clean up all my schedules of the week after Thanksgiving. And if you do study, besides eating turkey, and during the Thanksgiving, and feel free to stop by my office as much as possible, and I will be happy to go over some of the things with you. And so we covered a lot during this semester, and as I mentioned, the exam will be the closed book exam, the open book exam. Yeah, just make sure you're paying attention. Just to change my mind in the last minute. It will be open book exam, and so I will try my best to make sure that you won't find the answers in the slides. I don't know whether that's a good news or bad news. And but I also want to ensure you that you don't have to remember anything. Okay, I want you to understand things. If you understand it, you should be doing the exam well. And don't worry about the grades and things. I'm going to only fail half of you. So still half of you will survive. So don't worry about that. Okay, do a good job in the next week for the presentation. I know some of your your groups, the project actually started. That's impressive. But the presentation will be next week. It's on Friday. And I'm going to record the presentation as well. And I may have some fun to watch that during the Thanksgiving breaks. So again, if you have any question before or even after the exam, feel free to stop by. And I'm going to create a course evaluation in the on-course system, and you just say whatever you need to say. I'm not going to share that with anybody. It's just helping to improve the future lectures. And in your future research, especially if you're interested in doing next-generation sequencing using this technology, and I think my group is always here available to help, and do stop by and talk to us. And even though there's no cost, even though you don't have funds for the analysis or things like that, that will mean we are not going to analyze the data for you, but we can still contribute our brains in that. And so if you are a biologist, you are interested in using this technology, and I will prefer that we talk before you do the experiment. And most likely we can set up meetings with Dr. Edinburgh and other core facilities and to get through that. Okay? All right, so I will look forward to your talk next week. All right, thank you very much.